Neuroscience for allowing me to present my research today. So I'm not going to go through and bore you with my title again, because Dr. Sandstrom, introduced. Okay, so first of all, I want to introduce that as we all sit here in this room, all of us will age or are currently aging right now, and there are some problems that are associated with aging. Now, as we all know, first of all, we tend to lose skeletal muscle mass. And here in this image, what we have is an MRI cross-section of a quadricep muscle of a 25-year-old male and of a 65-year-old male. And both images here have this white dot in the middle representing bone, this dark mass representing skeletal muscle, and this white surrounding it is fat. And I want to point out, first of all, that the actual size of the quadriceps are about the same size in both the 25-year-old and the 65-year-old male. However, there is more fat that is surrounding the tissue as well as innervating in between the fibers. And so aging accounts for about 1/3 of the loss of skeletal muscle. And so aging is not specific only to skeletal muscle, though, but we also get problems with our nervous system. And here is a cartoon of a motor neuron that is from a healthy individual, and here is a cartoon of an aged motor neuron. And I want to point out that the aged motor neuron represents a smaller cell body size, where we tend to lose motor neurons when we age, as well as we tend to lose dendritic complexity. And so as we age too, the type of motor neurons that we lose first are fast type motor neurons. They're followed by our slow type. And by the time that we turn to be 60, we tend to lose about 50% of our motor units, which again, we lose those fast type motor units first, followed by our slow type. And then furthermore, by age 90, we will lose 70% of motor neurons. Now, one way that we can combat aging is with exercise. And here is Mr. Beefcake. And what we have here is some characteristics of what happens with skeletal muscle with different types of exercise. And so what he has is hypertrophy of skeletal muscle, or an increase in muscle mass and muscle fiber volume, as well as with certain types of exercise, we can actually get hyperplasia, or actual increase in skeletal muscle number. Now, exercise, again, just like with aging, is not specific only to skeletal muscle, but also with the nervous system. And there are some different adaptations that exercise can do for our nervous system. Here, what we can get is an increase in dendritic complexity with an increase in dendritic arbors. Here, we can get an increase in cell body size, as well as preservation of the motor neurons that we have. And also, exercise can induce protein synthesis and changes in gene expression. Here, exercise can induce changes in ion conduction, where sodium conductance is one known to change with exercise. Here at this point, what we have is a change in protein transport from the motor neuron cell body to skeletal muscle, as well as transfer of proteins from skeletal muscle to the motor neuron cell body. And then finally, we also get changes at the neuromuscular junction itself. And we can get an increase in nerve terminal branching at the neuromuscular junction, an increase in neurotransmitter release of acetylcholine, as well as hypertrophy of the neuromuscular junction. And some areas also have found that exercise can actually lead to changes in neurotrophic factor proteins that are expressed. And this is our particular area of interest for our research. The certain neurotrophic factor protein that we study is glial cell line-derived neurotrophic factor, or GDNF, as I'll call it from here on out. And why it's important is that to date, it's been found to be the most potent neurotrophic factor for motor neurons, where exogenous treatment with GDNF can actually rescue motor neurons that have undergone spinal cord injury. and can retain 100% of their original size. And furthermore, mice that are heterozygous knockouts for GDNF tend to lose about 22% of their motor neurons. And then furthermore, the homozygous knockout mice actually cannot develop, so suggesting a very important role for GDNF during development. Okay, independent researchers have found beneficial effects for exercise and for GDNF that are quite similar. However, nobody has been able to link the two. Exercise and GDNF can lead to hypertrophy of the neuromuscular junctions, can lead to changes in motor neuron morphology, as well as increased neurotransmitter release of acetylcholine. Now, some of our studies are trying to determine maybe some of the beneficial effects that we see with exercise might be in part driven by changes in GDNF protein content. And so what we did was look at exercise and levels of GDNF protein content in skeletal muscle. And with 2 weeks of exercise at a low intensity pace, what we found is in soleus muscle, which is a slow type skeletal muscle, we actually get an increase in GDNF protein content as compared to control. Now, one way that GDNF communicates is that it is produced by target tissues, and in our case, we're interested in skeletal muscle, and it is shipped back to the level of the spinal cord motor neuron, where it has local effects there. Now, as I previously have just shown, is that exercise can increase GDNF levels at skeletal muscle. However, my interest area is to determine if we get changes at the level of the motor neuron. If GDNF levels are comparable to those that we see with skeletal muscle, and if we get a similar shift with exercise. Okay, and that leads me to my hypothesis, where short-term exercise will increase GDNF protein content in rat spinal cord. Okay, so what we did was we looked at 3 different modalities of exercise. And we used aged animals that were 6 months old, as they were considered to be adult rats. And all animals were exercised for a total of 2 weeks. Our first group was a voluntary group, where animals had continuous access to running wheels, and they were monitored on a computer system that measured the intensity, the duration, and amount that animals would run per night. We had a second group that underwent involuntary exercise. And these animals ran a total of 2 hours, which was used to match the average amount of duration that the voluntary group ran. And these animals ran at 10 meters a minute. Now, this intensity was chosen because of my previous image where I showed you that we had changes with GDNF protein content in the skeletal muscle, and that was the same intensity used there, which again is considered to be a low-intensity walk training protocol. And then our third group that we used was a swimming protocol, where animals were placed in barrels and in water maintained at 37 degrees, and they swam a total duration of 2 hours, again just to match the running animals. And then finally, of course, we had control animals that were age-matched and sedentary and had absolutely no exposure to any type of exercise. Okay. At the completion of our studies, we removed the lumbar sections of the spinal cord, where we measured protein quantification for GDNF using an ELISA and Western blot, and then we used separate sections of the spinal cord to stain for immunohistochemistry. We used antibodies against GDNF, antibodies against choline acetyltransferase, or CAT, which is an enzyme that is a cholinergic marker for motor neurons, and it is an enzyme that synthesizes acetylcholine. And then finally, we used antibodies against calcitonin gene-related peptide, or CGRP, which is a neuropeptide produced at the level of the motor neuron and shipped to skeletal muscle upon demand, such as with development and with exercise. Okay, and so what we found was that, in fact, all types of exercise do increase GDNF protein content in the spinal cord. where here in this axis, what we have GDNF protein content here in picograms per milligram of tissue, and on the x-axis we have all our different exercise groups. Now, I did analyses to compare the exercise groups, but there was no significant difference between them. However, I would like to direct your attention to the voluntary group, where these animals would run at higher intensities, averaging about 18 to 21 meters a night, 21 meters a minute, whereas the involuntary group ran at an intensity of 10 meters a minute. And again, the involuntary was considered to be our slow intensity training. And again, with the involuntary, although it's not significant, there is this trend towards a higher amount of GDNF protein content with the low intensity group. But again, it's not significant. Okay, and here are our Western results, where in lanes 1 through lanes 4, we have different sample homogeneous of the spinal cord from voluntary animals. In lanes 5 through lane 7, we have different samples of control animals, and then lane 8 is just a negative control. And with these results, we see immediately that there is more GDNF content in the exercise groups as compared to controls, further confirming our results with our ELISA. Okay, and here are some histology slides. In panel A, this is a representative spinal cord section from a control animal. In panel B, this is a representative spinal cord section from an exercise animal. Both panels are stained in red for choline acetyltransferase, or CAT. Both panels are stained in green for GDNF, and then both are stained in blue for CGRP, with the purple or yellow as the overlay. And what these results immediately show us is that exercise does in fact increase the motor neuron cell body as compared to control. We also see that GDNF is localized to the region of the motor neuron. And I did not see a significant difference here with CGRP, although I have not done intensity staining yet. So I still have to look into that. Now, I did do analyses to compare the different exercise groups to see if there were actually, in fact, changes in motor neuron size. And we did find that all types of exercise had larger motor neurons as compared to control motor neurons. Now, I would like to direct your attention once again to my involuntary exercise group, where although it was not significantly different from any of the other exercise regimens, there was this trend towards an increase in size of motor neuron cell body size. And this was the same group that also had a trend toward the most GDNF protein content in spinal cord. Again, not significant, just noticing a trend. And so with that, I would just like to summarize that we did in fact find that 2 weeks of all types of exercise of involuntary training, voluntary training, and swimming all had an increase in GDNF protein content in the spinal cord, as well as an increase in size of the motor neuron. Now, there are some more questions that arise, of course, with any research project. And one area of interest I would like to look into is in situ hybridization, where the histology slide that I showed you earlier was for GDNF protein. However, we don't know what cell types exactly are making GDNF, and if there are changes in GDNF transcript with exercise. So if we're able to determine what cell types are making it, and if there are changes with exercise, that will help to understand what is happening as a mechanism with exercise. And then one other area that I'm currently looking into is to determine what is our optimal exercise program to increase GDNF protein content. Now, I've done some previous studies where I've looked at long intensity or long duration, high intensity, and have found that it does not have as good of an effect for GDNF for the motor neurons as the low intensity, short duration exercise. So again, if we're able to determine an optimal regimen, that will help us to understand nervous system plasticity. Okay, so with that, I'd like to conclude that if we are able to increase GDNF protein content with exercise, it may be one way of preventing motor neuron losses that we find with aging, with spinal cord injury, or with diseases such as with ALS or spinal muscular atrophy. So we need to keep exercising in order for our neurons to keep happy to talk to our skeletal muscle. Okay, so with that, I'd like to thank my husband, Matt, and my advisor, Dr. John Spitsbergen, my lab mates, Amy and John Mary, and then my committee members and funding from NIH and WMU. Okay, thank you. Any questions? Yeah.
So, Monica, that was a very nice presentation.
Thank you.
Two questions. First one is just a joke. Is that Dr. Spitsbergen in the picture running in the snow?
You know, it's not, but I tried to get one of them and I couldn't find one.
Oh, it's apparently him lifting the weights.
Yeah, that's actually him. It's identical, right?
So, this is a really nice model of those that go swimming or those that go walking, happy those that go running.
Right.
But one of the things as an aging individual I read a lot of is that weight training is really what we should be doing. Is there actually a model for the rat that you could perhaps use for lifting weights?
The one model that I've seen is for animals to climb ladders and to attach weights to their tails. That would be one way to induce weight training. Because unless you have like a little lever to raise their arms, I don't think they could do weightlifting. So.
That's what it's on.
Yeah, it's true. Yeah. Good.
Yeah, I'm just curious. It's my understanding that when these kinds of changes are taking place due to the exercise, that there's synaptogenesis and maybe even more synapses with the same motor neurons. And I'm wondering if it's your expectation that a large proportion of the increases in GDNF that come into these cells could be coming from the fact that they're making more contacts, or is it because it's being produced at a higher rate, or can you tell the difference?
Yeah, so that's why I think with in situ hybridization, we could see if the target tissue is producing more GDNF, or it could be surrounding glial cells, because astrocytes and Schwann cells are known to produce GDNF, as well as motor neurons themselves. So I think with that, it would be important to see what motor neuron, the fast type or the slow type, if there is a difference between the two in the amount of GDNF that's being produced, as well as determining what other cell types are producing GDNF. Okay.
That was a really nice presentation. GDNF might be one candidate trophic molecule. There could be others as well.
Right.
Have you looked, or others looked, in GDNF heterozygous knockouts to see what effect exercise has in preserving motor neuron size and muscle integrity?
I have not come across any papers that have looked at GDNF knockouts for exercise, but one area that we might look into is actually to neutralize GDNF in exercise animals. I've read IGF-1 is where they've looked at ALS models on mice, and they've seen beneficial effects of the with exercise there. Yeah. So good stuff. Okay. That's it for questions. Then I'm done. Thank you.